Dear students, good morning. Please put your attendance. Sarathi. Just a second. So we are going to continue with our earlier discussion where we are talking about the dosage forms, where clinical trials, what are the different stages. This PPT is already there. You can always consult it. I would suggest that you consult it before this end of this class. Rather, you can check it out. So basically, we were talking about the different stages in our last class, and I'll just finish it off another 5-10 minutes. Not more discussion on that, because we have another class for this clinical trial aspects later on in this course. So basically, when you are talking about drug development part, I think my screen is already shared. You can always check it out.
Sir?
Yes.
Sir, the PPT is like it is like reflected, sir. It is not in a proper way, sir.
Oh, I don't know. There is some new tools this Google Meet has introduced, and we are always confused. Let me see. It is
Now
what?
it is clear, sir.
My screen is clear, right?
Yes, sir. Now it is.
This is clear.
Yes, sir.
Oh, thank you. So in our last class, we already came to this point and we concluded that day. I'll just continue quickly. What are the different expectations from different stages? Discovery and development, mostly in research lab, preclinical research, also research lab and industry. Clinical research, all those things are definitely from industrial perspective. Who is going to put these formulations into market? Some FDA review will be there, and finally post-marketing review. Once the drug is already out in the market, these things should be implemented. Whole process can take some drug development, at least until this FDA review process can go some 10 to 15 years or so. Exception is always there. And finally, this can go lifelong, infinity time period. Maybe the safety review can continue for certain drugs. Even some drug has been withdrawn after 30 to 40 years after they are in market. So this study is really very important when you are considering the safety of these drugs for individuals. I think I already talked about the investigation on the new drug when we are going to do for drug development things. Product from the target product profile through post-approval activities is there, usually prepared prospectively. I already told prospective study, retrospective study, right? Most of the time, the drug analysis and clinical trial will be on prospective basis. And it is definitely a logical stepwise fashion will go. So preclinical stages for drug development will be the discovery part, toxicity, manufacturing, and stability. These are all coming, these are all background study. They will come before the clinical trials really happens. Discovery part is all mostly in academic research lab, and of course in R&D centers for different big drug houses, they also do. So they are actually looking for the basic research part. They understand the process, how this is going to control the disease, cellular, molecular level. So laboratory studies, maybe in cell lines, maybe in vitro studies, and some cases in vivo studies in animal models can also be done. But this is all in academic research lab they primarily focus on. So they are finding insight into the disease process, the pathway, and what could be the target molecule. Even many of your maybe your seniors or friends who has done the projects, they can also identify certain molecules for a disease, because pathophysiology, how the disease works, is mostly known to us. So if you know it already, it is all good. If you don't know, you need to know the pathophysiology of the disease, how the disease happens, what happens physiologically, what happens in the metabolic pathway, changes is there or not. So understanding the disease and its effect, what changes is happening directly or indirectly, and what is the cause of this, what is the root cause of the disease state, manifestation of the disease. These things need to be studied. Once you know this, at least, okay, this is the disease, this is the effect, if I block this, if I can stop this, then it can be served as medicine. This information is acquired. We need to know those essential things. So essentially we know what is going to happen. Here one example is given, right? A gene and protein is essential for a disease process. So if we know what gene is involved, then possibly we can go for treating those expression of those particular gene, or blocking some essential receptor on this pathway. So that means I know what happens in the disease state. Maybe it is leading to one molecule, it is binding with something else, and this is blocking the signal transduction. So you may block any one of these routes if there is any receptor for binding these domains, and you can look for some molecule which will bind there. That means this pathway can be at the very beginning or at the terminal before the effector stage. Anywhere you know these are the molecules which are involved or receptors, you can block. So essentially understanding the gene expression study, understanding the disease, understanding the mechanism, mostly done through literature review and academic research institutions or clinical research institutions. They can also do those study in laboratory scale. And once you know the pathway, once you know the disease, how is it going to happen, so we need to find out how can we go for a compound which can have a beneficial effect. Many people do these drug discovery components using your bioinformatics tool. Okay, so molecule identifications from a large database, okay, where you can have some million number of molecules, sort to larger molecular weight or high molecular weight molecules are there. So some work they can do in computational domain. Okay, so they are going for whether this molecule is binding it or not with your target site, the binding is stable enough or not, binding is favorable or not, those physiochemical properties of the molecule, and the receptor binding domain, what are the amino acid involved there, for example. Those studies they can always do in a molecular docking studies. Okay, so they can find out possibility. Once they know the number of molecules, they can screen it out and do some other studies. So once the molecule is ready, or maybe number of molecules are identified, these are called lead. So based on these lead molecules, further chemical synthesis can be done, and they can identify one or two compounds which is possibly having the physiochemical property like solubility. Many properties required. Once you are identifying a drug, many properties is required. What is its solubility? How much it will be adsorbed? What is the rate of elimination? What is the stability? Many questions comes, and they have to, like a checklist, right? So they will have, okay, everything is okay. Okay, once this criteria is fulfilled, then this lead molecule can be a candidate for clinical trial. And before going to clinical trial, they need to identify whether this is going to be any beneficial effect or not. That is the pharmacological effect. Drug, when it is administered, what is the effect we are observing, right? Cause and effect. You are administering drug, what is the result? So they need to find out whether the intended result is coming or not, whether it is beneficial for this or it is not going to be beneficial. Is there existing treatment that have any unanticipated effect that we never know, but some other effect is happening? That is, we will say it is toxicity kind of things, or untoward or side effect rather. So apart from the original intended effect, the drug is going everywhere, right? You cannot really focus. You are not always using targeted drug delivery, but in some other organ, the drug can have some wrong consequences you need to know. And how can we have the new technologies that provide new ways to target medical products for specific site? If you are going for targeted drug delivery, you need to know where it is going to work, but always it is not possible within the body or to manipulate the genetic material. So these things are not widely used. Genetic manipulation of the material is not widely used, particularly for chemical formulations. Some bioformulations may be there. So in some cases, they are going to have the vector-based replacement of the genes for a particular organ. So these engineering aspects are always there, but we are mostly talking about pharmaceutics. So dosage form, which is essentially a chemical molecule, small to medium molecular weight. So traditional drugs we are talking about. So this may not be involved. And it can take some time depending on how they are collaborating, some 2 to 10 years. That may require to complete all those aspects of the study. These are all coming before you are going to select the molecules. So your selection of molecule is nearly confirmed and you are going for preclinical testing. So that means you probably look for some animal testing here. You see the novel drug products undergo laboratories and animal testings to answer some basic questions. Animals can be depending on what kind of drug you are going to utilize. So you can have the animal models. Okay, you can have mice, you can have guinea pig, or many more, depending on what route of you are going to administer, what is the effect you are looking for a drug for systematic circulation, and how is it going to affect on the individual. Those queries may be there. So we are going to have using some computerized model also. We are not going to do some kind. We will look for cell, maybe in animal tissue culture lab also. The person who is looking for the changes in physiological state. For example, I'm going to look for activity of a drug on liver cell. So we can have a cell line for a liver cell line. We are growing in a lab and we are adding the drug or pharmaceutical compound, and we are going to see the morphological change, physiological change, or expression of some enzymes. For example, we are going for the enzymes which is produced by the liver cell. We are going to check. So essentially, you can do some amount of study in a cellular level using your lab. Academic lab is maybe appropriate for this. And animal studies, you probably know in our university also we are having animal house, and there is a ethical clearance committee is also there. So you can undergo some studies for a long to short term, depending on what kind of animals you are going to utilize. We can have animal testing facilities. We can include the animal in our study design. And depending on at what pace you are doing, how much data you are collecting, it can take 3 to 6 years. So you understand, before you are reaching preclinical studies, you minimum have, for example, 5 years you have already spent, 5 to 10 years you have already sacrificed. That is this is actually coming up before you are going for anything on clinical trial. If you are fail here, then your money is gone, everything is gone. There is no use on that. If you are successful, then you will definitely proceed to the approvals for new drug. You will be submitting the request for review on animal studies. I mean to say human studies, sorry. Human exposure. If I'm going to have the request, you are going to put on FDA. So you may have some request for exemptions from FDA to ship an unapproved product. That is different part. That is a country specific requirement maybe also be there. And we need to have the investigator need to put the applications, how we are going to go for this data collection process. So before you are applying, you need to put key elements. When I'm going to have the human trial studies, should include considerations for product activity, ability to replace. That means if there is existing some drug, how you are going to be better one. Ability to replicate, whether can you do the study, how many study have you done. For example, you are talking about animal models, how many animals you have studied, how many times you have studied. So these are the basic requirement that will be judged. So this is called proof of concept scientific data, what you have already gone in your lab, animal models, cell lines, how is it going to replicate, with reasonable explanation how the product will prevent. If I'm going to have some treatment purpose, if it is a diagnostic kits, you can treat or cure diseases. So this explanatory note is definitely required. A target clinical indications for these drugs, what is your plan and protocols, what you are planning to undertake. Animal toxicology data, if you have done this study in animal models. Manufacturing process, what kind of formulations you are going to undertake if the drug is there, how it can be implemented, including analytical test result confirming the quality of the products in several batches of the product when they are going for look for pharmaceutical formulation. Okay, so finally it will come as a formulation. How is the drug stability studies? So this is actually physiochemical study under different storage conditions when you are going to use in the period of use of the clinical trials. So these informations may be required when we are going to prepare for this. Okay, so this is not necessarily for any research lab, but for example, when your pharmaceutical industry is looking for how they're going to do for quality guidelines or good laboratory practices, when we are having non-clinical laboratories, there is some reference guideline, you may always check for that. And when I'm coming to clinical trial stage, it can go longer, 6 to 8 years we are projecting, but depending on, it may also be time period may exceed in some cases. And when we are going for, we are going to see what are the things we are going to include, which should be included. Someone may be the sponsoring is the pharmaceutical giants where they're going to do. And here site or PI, when I'm going to have a principal investigator, for example, I am the researcher, someone is sponsoring. So these things should be adjusted, they should be finalized when I'm going for a clinical research studies or clinical trials should be conducted on that. So we'll have site selection where you are going to do the study. I mean to say what is your area of study where you are going to do. Confidentiality agreements, that is actually minimum requirement that whatever data you are going to get from those individuals, that confidentiality should be mentioned. Feasibility, how many numbers are there, feasibility assessment should be there, and it should be taken care by both the groups. Feasibility in terms of the number of individual, feasibility in terms of the population you are going to collect, and whether the analysis will be statistically valid or not. Legal and financial activities, anyway, we need not bother on these aspects. That will be taken care by those sponsors over here. We need to collect the regulatory document, who is going to look for site and training, monitoring and study execution, study closeout. These reports are primarily for the researcher part, who is going to conduct this research, data analysis, and you are going to finally submitting it for the approval process. So you see, when you are going to have these different phases, phase 0, phase 1 to phase 4, primarily 1, 2, 3, 4 is the more involvement here, and we will just go what we are going to see in this study. So when I'm going to say phase 0, we are going to go for microdosing. We are not really going to treat some individuals. We are going to give it for very short period of time and very few number of people. Of course, consent, all those things are always there. Does it affect the target or not? So this is very big questions before you are implementing in a large group. You cannot really sacrifice people. Okay, this is not desirable, and you cannot do that. So some very small subset of population they're going to go and do for a small duration of time. This is primarily intended to get information whether it can really be included in the study design. So subtherapeutic dose to obtain the preliminary pharmacokinetic and pharmacodynamic data in individual. I will talk about the pharmacokinetics things and pharmacodynamic things later on. Just for time being, let me give very small brief intro on the pharmacokinetics things. Pharmacokinetics is you are an individual, right? You are taking a drug, and once you take the drug, what is the concentration of drug in your bloodstream? For example, we are monitoring and then coming out. So this is actually pharmacokinetic. This is called pharmacokinetic data. Okay. Pharmacodynamic data is something different. It is the result. Result means, for example, for example, you are taking some analgesic and there is an indicator of the pain, extent of pain once you take the drug. So there will be some drug doses you are taking and you will have a result like that. After that, even if you take more dose, you cannot see more improvement like that. So this is actually drug dose and result. So this is a dynamics of a molecules. So I'm increasing dose after a certain time, no further benefits. This study will be done. So what could be the possible maximum dose I should give? And this is kinetics, that is the concentration in blood. One tablet you just popped in and you are going to see, this is time, of course, this is time. So with time, how the blood level is increasing, I mean to say, and decreasing, those studies are essential. Kinetics, dynamics. We'll speak on them sometime later. So these things are essentially required when you are going for a drug to be included in zero clinical trial. Some data, of course, you cannot do the entire study on this. Gives no safety and efficacy information here. It is very small group, just to see, allow the products to be evaluated on human data instead of animal. That is the first thing you are going to do. Support decision to go forward or not. This is very, very important. No therapeutic intent. We are not going to really see whether finally we are going to have the drug or will be effective or not. Not like that. First preliminary, for example, 7 people I am administering or 10 people I am administering, and people are live at least. So the principle of this study is to microdosing for very small group. If they are all healthy, all are not dying out, you cannot do with 100 or 1,000 people. You cannot risk. So this is actually the initial stage to screen out whether we should proceed into larger population, phase 1 or phase 2, phase 3, all those trials we are going to look for this or not. Now, my primary intention in this class, when I'm talking about phase 1 clinical trial, what we are going to do, what is the questions we are going to answer? Is this drug candidate safe? You see, safety is different and result is different. Is this drug useful or not? That is not the questions. They're primarily looking for, is this drug candidate safe or not? Safe or not means we need to know pharmacokinetics also. How much dosage it is coming, whether this dose is causing any toxic effect in the individual or not. So these informations, I don't know, some problem in the tip. These informations need to be addressed, and we need to see whether this is there any safety, toxicity events is happening or not. Short term, this study is mostly conducted in a short term period. A population is in a moderate number, 20 to 50 normal healthy individuals, volunteers, who is going to have. Occasionally, where the drug is to be used for patient with a particular disease, we can also include those subjects. So if you know this drug is for certain disease you are going to intend, you can take. Study design is to escalation of the studies to establish maximum tolerable dose. That means what is the maximum dose we can give an individual beyond which toxicity is not seen. Always try to understand, when I'm putting any drug, there is a 2 concentration line. One is known as minimum effective concentration. And here, minimum toxicity concentration. What is required when you are injecting any drug or you are taking one tablet or capsules? The blood concentration of the drug will increase, and okay. So until this period, you see, concentration is below the minimum effective. So this period, nothing, you will not see any benefit. Similarly, in this period also, below MEC, no, you will not see any effect of the drug. Drug is there, no use. Only this period, it is above the MEC, so you will see the response is happening. Similarly, if you take some other dose, you may reach toxicity level and coming down. For this thing also, you will only experience anything good between these two periods. Only this small hour, you will have any benefit. And similarly, during this small time period only, it is beneficial for you because it is in between MEC and MTC. Above this means it is already toxic. You are already experiencing some toxicity. So drug concentration is to be really carefully managed. You cannot get this by simple theory, right? You have to do the study itself. How much dose is reaching and what toxicity you are observing. And then only you can draw the line. Otherwise, you cannot draw. So this study is already done. Once you have this, we can establish the maximum tolerable dose also. This is minimum. This is minimum for toxicity part. We can also say that this is toxicity is coming up means we can also put the boundary here. We should not go beyond that. If you are going beyond, we can also put a maximum limit. Maximum limit is actually done some on theoretical basis. Okay, normally 80 and 20% ratio is already put. So we can put some 20% allowance. So for example, I'm getting some 30 milligram per liter, just for example. You can take some 20% allowance of that plus 20% of this. You can put some margin. Okay, beyond any situation, any circumstances, drugs should not go beyond that. That can be potentially lethal for the individual. So focus of the study is safety and tolerability on human. So how can you do that? By, as I told you, pharmacokinetics, bioavailability I'll talk, bioequivalence also I'll talk, bioavailability and bioequivalence. Bioavailability means how much is coming into your body. That is very important. How much is bioavailable? Okay, you are taking, for example, let me tell you, you are taking a drug which contains 100 milligram, but this is no use until it is coming into blood. So if the drug is not available to your system, if your system cannot absorb the molecules, for example, I'm taking popping up a tablet, so mostly the drug will be absorbed in small intestine, mostly. Otherwise in stomach, the condition is too acidic, so a drug can be ionized form and you cannot really utilize them. So how much percentage of the drug you are actually absorbing? It will take some time because in small intestine, the drug molecule has to break down, they has to dissolve, and blood is already circulating, right, on the lumen. So in the lumen, while this drug molecule can penetrate, drug should not be ionized, otherwise it cannot cross the barrier. Many questions are there. So out of your molecule, how much molecule is actually going inside your body? That is known as bioavailability. So you may take 100 milligram, but actually blood, it is reaching only 70 milligram. 20 milligram is simply excreted, okay, with stool, for example. So that is bioavailability. It's very important because it will also decide the dose. Bioequivalence is I'm having 2 tablets. You know, na? I can have some, for example, paracetamol as a injection also. I can have paracetamol as a tablet also. I can have in some suspension also. Do you think all of them will work together? No. To make them equivalent, I have to change the dose. When I'm injecting, I need less dose. When I'm putting in a suspension formulation, it will take a little bit less dose because it is already disintegrated. If I'm taking tablet, I need to consider that tablet will break out or not. So equivalence means different dose. The result, the outcome should be similar. Is it there or not? Then they are called bioequivalence. Biologically, their outcome is similar. So maybe I have to put somewhere more, somewhere less, but final outcome will be identical. So that is called bioequivalence. Metabolism and toxicity study can be done. Second, here only we will look for whether drug is working or not. That is called drug response. So we will look for a little bit more, a few hundred volunteers to target population. Here, result is more important. Whether we are going to get the result what we are looking for. What design? Randomized blinded trial. Blinded trial, I already told you, blinded means the person will never know whether what she got or he got the drug correctly or not. This is important. So blinded trial is conducted. Strict eligibility criteria. So we should definitely define who will be considered in this test or not. Otherwise, we can actually exclude them from the study design. Usually not compared to standard of care therapy. We are not going to do this. Whether we are comparing with the existing study, no, not like that. It is only for the drug, whether it is effective for the desired purpose, not undesirable. Dose response, essentially. If I put the drug, how much response we are getting physically or not. This is very important aspects. So this dose estimates for subsequent studies, we will find out what dose is ideal for this candidate to have the established effect. So effectiveness of a specific population in a disease, collect additional safety information if you can have. Finally, outcome is minimum effective dose. So what is the minimum dose I must give? If I take less dose, no result will come. So you please understand, small amount of concentration, if I'm injecting lifelong also, no effect will be there. So we need to have a minimal concentration beyond which any effect is there. Okay. So for example, someone may say, I'm having one dose of drug of 600 milligram, for example, paracetamol is there. You are thinking, no, 600 milligram per day. Better I will take 200 milligram, 200 milligram, 200 milligram, small capsule. Every 8 hours I will take, sir. No, you will not get the result because the blood concentration of your drug will be always very low. You are taking same amount. The total drug also going into your body may be same, but your concentration is always sub-optimal dose. So you will have no effect on this. So concentration and dose of this drug is very important when you are going to have the clinical trial phase 2, now phase 3. This is very important, and this trial is this drug candidate more effective than any existing option? Is there any treatment already available? Yes, some treatment will be always available. We can always offer some suggestions. So phase 3 clinical trials is going to pivotals here. Pivotal means very important in this design, and they can be considering what is the advantage we can have. Parallel anticipated treatments often can be years. So how much long we can going to do? 100 to 1000 population can be in this case, in this treatment process. It is also expanded randomized double-blinded control trial. Double-blinded is very, very important. Double-blinded means I am the researcher, right? For example, you are the clinical trial subject, test candidate. I also do not know what I am giving you. You also do not know how much you are getting the drug. Then you say, how is it possible? Oh, okay, there is something is there, something is recorded definitely. So we are recording all those data is actually recorded by a numeric code. So that code is already prepared by the administrator. So some administrating agency is going to do this job for your research purpose. You are the researcher one, this is the test subject. So with whom, what drug is added, what is the concentration I am going to give, that record is there, but that they know, you don't know. You will simply give the number. So test number, for example, some ID is there, and this drug you are administering. So that means you are also blinded from what doses you are going to give. The design is there in the background, but the administrator do not know. What is the benefit? Because biasness in the study design can be avoided. If you know something, because you are a researcher, something will come in your mind, okay, I'll give only the high good dose only to get the result. So to avoid this biasness, you can have sometime double-blinded control studies is there. Here we will focus primarily how is it going to affect in comparison to other molecules, the risk and benefit analysis, whether it is really worthy of having the drug, already some drug is already there, dosing interval, anything labeling information means how much dose you are going to give, how to provide the dose, and any safety profiles if you are going to study. So until this point, most of the study, what is required for your drug discovery and coming into market, safety, all those things are all over. Okay, these you have already done. So basically, if you are passing all those things with flying color, that means very likely it can be considered for advancement and it is approved. And if it is approved, there is also further studies you are promising to make is post-market approvals. How does the new drug perform in general use over time? Over time is very, very important. 1 day, 10 day, taking something may not give you any effect until or unless it is going to accumulate in your body. So long-term effect can be something different, which you are not expecting out of your formulation. And that can take some years, not only 1 or 2 years, maybe some 10, 20, 30 years period may be also be required. So what is the focus? Any safety issues, if it is coming there, or efficacy issues, some cases you never know. Okay, after a long time you are finding that, okay, if I am taking this drug with some food, effect is not there. So that means you cannot really study all combinations, right? Whole population in the world is having different habits, different food habits. They're having different physiological stata. You cannot really check each and every one before you are allowing the drug, then it will never come. So long term, once you are going for a study, you are just recording the informations. Some reporting is there and they will get back to you. So it is like a feedback mechanism, so that these things can be further subjected to deliberations and they will find out if there is any rare side effect. Rare, very few people it will be having. So rare means I need to have some 100 and thousands people. Okay, even more than that. So large group of population can be involved there, which is already there in most of the drug. And any age and ethnic groups if you may involve, you may consider that particularly cases for your study if it is required. And finally, feedback from the patient part if you are going to have any formulations. So basically, okay, I think that is sufficient for this discussion. Other part is not really for our discussion part. We will not consider on that. So clinical trial, the different phases, if you just summarize and you can see what we are looking for. 0 is for small population, just to be safe that people will not die out, right? People are not animals. Like animal study you are doing, you cannot sacrifice. First one is PK, safety and toxicity. First you are talking about these toxicity and safety. If these are coming out, if this is not so severe, go for dose response. How much dose I should take for making a big group. So dose response is coming into 2. Third is the most important and you are going to get the result out of that. So what is the effect, how long it is going to take, and you are going a big group, blindfolded clinical trial studies, risk and benefit, dosing interval. Most of the questions will be addressed here. Okay. And you can see the last one, which is beyond the scope. That means we have already covered up these drug molecules coming into picture and market. How can you deal with those outcome? How can they take back the feedback and adjust the dosing regimen if it is required? So I think I will further component is not really we are not going to discuss here. You can go through. So that is the first part we wanted to discuss in this class. And okay, next components what we are likely to discuss today. Just I'll start. We're not really likely to discuss. Yeah, that is, I think I remember, this is drug. Sorry. Dissolution and absorptions we are going to look for. We are looking for chapter 5. I will just give you the chapter 5 outlook. Everything you cannot really study on the same day. It is a very big textbook, so you need to be very focused on that. So what we are looking for is how the regulatory requirement, there are many other components are also there. We are looking for the stability of the compounds, if we are looking for on that. Just a second. Sorry, chapter 5, not, I think I have written 4, 5 only, yeah. So here we are talking about dissolution, absorptions, bioequivalent testings, those things we will discuss. You may please go through this chapter to some extent, not everything is required. Some other things we will be discussing in coming days. Just we will talk about the dissolutions for this day. Okay. What is my topic? So we'll be talking here itself. So dissolution is not really relevant for everyone. All pharmaceutical dosage form really don't need these aspects. I just need to talk on this for some time. Dissolution and bioavailability. Let me start this discussion for a tablet dosage form. There are many dosage form, right? You have already seen many of them. So dissolution comes into picture for mostly on tablets. You know what are the ingredients of a tablet should have? First of all, you need to understand. Of course, you are having some active ingredient, right? We normally say AI or the drug molecules. That is essential. So for example, this is a tablet. You have seen many tablets, paracetamol 500 milligram kind of things is written. But have you checked the tablet weight is more than 500? If you take this tablet and put in a weighing balance, you may find that, okay, this is more than 1 gram, 1.2 gram or like that. But it is written 500 milligram, for example, some paracetamol. So what happens? So paracetamol molecule is there definitely for 500 milligram, but there is some other materials are also there. These are known as excipients, additional things. Yes, these are all additional things. Excipients can be, what kind of things are there? First one is known as filler. Filler means these fillers are not actually any pharmacological compound. They have no medicinal use. Just to make a tablet, right? I want to have 10 milligram, some maybe many times you have seen those who are taking medicines for blood pressure management, they're having some 10 milligram. 10 milligram, can you make a tablet? So small. So to make it manageable size, I need to add something so that it will be a little bit bigger. It is more handleable. So filler is a large volume of material. It can be non-active. Mostly we use some starch or cellulosic material. This is the primary component for filling things. So filler is large volume. To make it volume, filler. There are no medicinal activity on that. So that is there. So filling is one of the important consideration when I am having there. We can also have some binder. What is binder? Binder is actually putting a glue. Okay, so for example, I'm having some drug powder. I'm having these starch or whatever you are adding. How to bind them so that they will be compacted into a tablet and they will not simply break out. So we need some binding agents or binder. We also need some time so that this tablet will actually break open. If it is too much binding is there, you are taking the drug and it will not dissolve at all. So we need something which can actually break it open, and this is known as disintegrator. What is disintegrator? Such chemical compound which if it is coming in presence of water, they will swell out, they will be little fluffy. We can have many formulations. We can have some polymeric material. We can have some, for example, effervescent tablets are there, right? So in acidic environment, they will produce calcium carbonate. I'm sorry, carbon dioxide will be generated. So we can have bicarbonate molecules. Okay, sodium bicarbonate. We can put some molecules on that. So once the water is coming, they will form some microbubble. You have seen, if I put a tablet in a glass of water, you have seen, outside surface is eroded slowly and then quickly breaks up because there is a presence of disintegrator. There is other ingredients also for preparing tablet, like gliding agents, which will move it, because tablet, I don't know, I definitely have not seen how the tablet is processed in a pharmaceutical industry. We were having in a training, so we know how they really do. So all those things, all the mixing, blending components are mixed, okay, powder form. They are all mixed in different proportion, and the whole powder is moving on a tray, okay, in a tray form. It is moving continuously. Now, if this formulation is not gliding, it is not going through a surface, then what will happen? Proper dose may not be filled, right? You cannot fill a proper dose in a tabling machine. So they are having a punch, tablet punch, or it is normally called dye is there. So it is continuously free flowing, and once it is filled up, this is on a chain, it will move up. And from upper side also one punch is there, which is going to press against this device. So you will have a tablet which will be rotated out in the next, it is a circular platform on which this thing is happening. So we need to have something which will aid in moving. So for preparation purpose, we need to have some gliding agent like talc, right? We put some powder. Okay, of course it should be pharmaceutical powder, like magnesium salt. We can put some amount. So there will be gliding agents. So they will not stick, they will move very smoothly, freely. Those things are also required. So basically, once we are talking about tablets, there are many components involved there. And when you are talking about dissolution, we need to understand, first it will disintegrate. Disintegrate means if I am putting into water, they will form, so for example, this is a tablet. It will be broken down into first it will be broken down into large chunks. And then we have de-aggregate. So if the chunk come together, then again they will be compact only, right? So we need to have some disaggregation. This is disintegration. Integration means breaking down. Aggregation means let not come together. So these will be again coming out, popping out, breaking down into further small particles. And they will not come into close contacts. So maybe we need to include those things in our formulation. And they will be all very fine particles generated. And as the fine particles is there, they will be slowly dissolved. If they are soluble, they will be dissolved in water, if solubility is there. So basically, dissolution talks about how they will be dissolved in a given fluid from breaking down into small part to solubilization. And this is very, very important because most of the time it also takes some times. Because dissolution, until it is in a water, it is not dissolved completely, it cannot be absorbed by your blood. Because you know, once you are taking this drug and it is going into your stomach part, for example, where pH can be very low, it can be around 2 to 3 pH, acidic pH, and it does not get much time. So your tablet may not be completely broken down. It's still some big chunk is there. Then you will have, for example, small intestine, if I'm talking about. So here in the intestinal wall, you will have the blood perfusion. Blood perfusion means blood is pumped on the membrane side. So your molecule, once it is inside water, they can cross this membrane, okay, and they can come into the bloodstream. So this crossing requires there is stringent requirement. It must be dissolved in water. So dissolution, at what rate they are going to be dissolved, that study should be done. So if you are doing a dissolution study, what you will do? I will take a tablet, okay, and for example, I am making a small agitation. Don't make it very big agitation. Some fluid movement is there. Okay, normally you do not put this this thing. Rather, for example, we are rotating at some 50 rpm or 60 rpm. So what will happen? It will disintegrate and slowly it will dissolve. So the liquid phase concentration of the drug, if we measure, you will see slowly it is getting dissolved, saturated. So this phenomena is known as dissolution. How much time it is taking? This time is known as dissolution time. So you need to understand that your body, your stomach, they also have some residence time, right? There is a valve here also. You know, I'm not very good in this anatomical structure design part. You can simply think about. There is also some time gap that is known as gastric emptying time. So here, actually, this is actually mixing. It is kind of mixy, right? This valve is closed, and there is a rhythmic process. So it is actually crushing down the food material, and then after some time, the valve will open. It will come. So you need to understand your dissolution time may be more than the time it is getting here. So what will happen? It will not be dissolved. So you need to have your drug which should be broken down, disintegrated before it is actually coming into your small intestinal part. Breaking down is required. Once it is dissolved, then only it can be absorbed. And it is flowing continuously, right? So you have to take the drug before it is coming out into large intestine. Okay, it is not possible. So we need to have a gastric emptying rate. We need to consider, we need to have the dissolution done before it is going to reach. So anyway, the dissolution process should be improved for any pharmaceutical dosage form. And if it is not dissolved completely and not absorbed, but rather it is coming with your stool, so that part is wastage only. So how much is actually you are absorbing, that is very important in your blood. So even if I took 500 milligram tablet, actually blood actually maybe some 300 milligram is coming. 200 milligram is simply wastage. So this is called bioavailability. How much you are going to get out of your formulation dose into your bloodstream. My time is over today. I have to stop it here right now. We'll talk in our next class. Any questions from any one of you? I know you will not have any questions. You don't ask anything. Still, if you have anything to discuss. Students, are you online or all of you have left?